Hello, my name is Alexander Mironov. I'm the manager of Electron Microscopy core facility in the University of Manchester. Today, I'm presenting another lecture from our seminar series Wednesday EM Talks that covers principles and methods of electron microscopy that are used in our facility. Today's topic is three-dimensional electron microscopy. What are the options? It will be delivered by David Smith, who is our youngest staff member and very promising young man.
Hi, my name is David Smith. I'm a technician here at the Electron Microscopy core facility for the FMBH. This talk, we're going to talk about three-dimensional electron microscopy, and I'm going to try and take you through what it is, and what are the options, and where the technologies are, and a bit of history. So this is like a lovely slide just showing some models, which is kind of like the end goal of a lot of 3D electron microscopy. You do your study and then you end up with a lovely figure like this in your paper, which is really eye-catching and also can be very informative. We're going to start right at the beginning. So in this talk, you'll hear me refer to the 3D data a lot, and I just want to be clear about like what exactly I mean by 3D data. So all the techniques I'm going to talk about today produce this 3D electron microscopy data, and what that means in practice is a 3D volume that was made up of 2D slices. These slices are in grayscale because they're electron microscopy images and there's no color. So if you look on the left here, this left panel, this is the slices viewed from above, and every time I go through a slice, the corresponding slice is shown on the right. So if you can imagine, as we travel through this data set, we're going slice by slice by slice. So these 3D data sets are simply collections of 2D slices that are placed along the z-axis. And the different techniques create different types of data, and the data varies in terms of like how big the data set is, how many slices, what's the distance between each slice, what the resolution is, whether the data set was taken with an SEM or a TEM, how big is each pixel, and also other things like the skill and time required. And all of these things go into like helping you to choose like what type of 3D data do I want to get, and how achievable is that with any individual technique. So this is just to show the workflow of 3D electron microscopy. So at the top here, you have your sample preparation, and you can go quite in depth on this. It can be quite complicated, but we're going to keep it very simple. We're not going to go over that today in too much detail, but as generally we're talking heavy metal staining and embedding in plastic resin. And the bit we're interested in and we're going to talk about today is this group of techniques here, plus a couple of other ones. And then after that, whichever technique you use, you end up with this raw data set of incremental z-planes, the 3D data set, as another way of saying it. And then you have your post-processing, filtering, making it nice for analysis, and then you have your reconstruction, modeling, statistical analysis, whatever you want to do, and hopefully you end up using it in a publication. But yeah, so the bit we're going to talk about in this talk is this bit here, a collection of different actual microscopy techniques. So we're going to start with serial section transmission electron microscopy, and the story of that technique is the same as the story of 3D biological electron microscopy in general. It's basically this idea of taking traditional transmission electron micrographs, which are projection of shining electrons through a thin slice of sample, and simply making slices that are taking a series of slices through the sample. These are called serial sections, and then imaging each one of those slices, and then modeling them and finding a way to kind of like create, kind of bring them together in a kind of by creating a model or something like that. So yeah, ever since people have been able to use electron microscopes for biological purposes, they've been trying to get some 3D information out of it. So on the figure to the right here, I think this paper's from, does it say? Yeah, 1957. What they've done here is they've taken some slices through a tissue, and you can see some mitochondria here. And then they've imaged each one on a transmission electron microscope, and I think they've traced around the micrographs on tracing paper and then layered them on top of each other, which is a very, very rudimentary way of doing some 3D modeling. Obviously, things have come a long way since then, but the basis of the technique is the same. So this is a technique that has been like very groundbreaking for its time, and it's still, I think, sometimes used today, but it's very much fallen out of favor compared to the techniques we'll talk about. And that's for reasons we'll discuss. But yeah, essentially what you do is you use a microtome, which if no one's done a microtome, just think of it as a very sophisticated knife that cuts very thin sections. You'd stain your samples and embed them. Then you'd do your sectioning on ultramicrotome. So you take section after section after section, and each section might only be 50 nanometers thick. And each of these sections is stored on a grid, and then you image each of these sections separately. And then you, in some way, you combine them, analyze them, align them, and then you create a model. If anyone's ever taken any sections on a transmission on a microtome, then they will know how difficult it is to even get one section well preserved. So to do it for dozens, hundreds, even thousands of sections is quite impressive.
Here is a data set from the CADL lab. This is some embryonic tendon, and you can see as we go through slice by slice, one thing to look at is like the data set is quite high resolution, and a good benchmark for that is that you can like clearly see the double membrane of the nucleus, and you can really clearly resolve your collagen fibrils and loads of other nice details of the cell. But one thing you're bound to notice is that it's quite jittery, and there are like black holes and things like that. And that's just part of this technique, because it's really prone to artifacts. Because if you imagine you're having to collect each slice individually, store it and image it, you're inevitably going to get some, damage to the slices, just because they're so thin. They're often 50 nanometers. These are very tiny things we're talking about. And here on the right, you have a model that they reconstructed. So these colored bundles are different unique bundles of collagen fibrils through the tissue. So some take homes about this technique. So it offers very high XY resolution. So that's the resolution of the actual images that you take. Because you're using a TEM, it's bound to be a high resolution technique. But the Z resolution, as in the distance between slices, is quite high relatively. We're still talking only 50 nanometers or something like that. But yeah, so your resolution in Z is lower than XY. Another problem with this technique is it's really prone to artifacts, as I've said. You can't do a huge volume. You're limited to the field of view of the microscope compared to other techniques. And a big downside, it takes a lot of time, and it's hugely labor intensive and very highly skilled. Anyone who's done this deserves props, because it's a big commitment. You also have this alignment problem, which comes from the fact that you have to manually align each slice. If you have this, obviously you won't have a banana in your data set, but if you have something analogous to it running through the Z planes, all you're going to see when you come to align this banana is the actual tops and bottoms of the slices. And as a result, you're going to naturally align the banana to be straight, which you can think that of an analogy to any, like a bundle of fibers or something like that. Yeah, that's an artifact that's introduced by this. But the limitations of this technique did not stop people from doing really great things with it. So this is a paper from 1984 where in 8,000 different images on a microscope, they managed to do an entire C. elegans worm and reconstruct its entire nervous system, which is a crazy feat. And I dread to think the amount of hours and dedication that went into that, but it's seriously impressive. Yeah, very cool. And it shows the power of this technique if you can use it. But obviously it has downsides, and that's why it's fallen out of favor in recent years. Because of the techniques that we'll introduce today that are a lot more modern and up to date, and all of them are easier to do. So the first one that we'll talk about is serial block phase scanning electron microscopy. And this is the one that I work on mainly here. This is a technique that has a lot of similarities to serial section transmission electron microscopy. But the big difference is, whereas with serial section transmission electron microscopy, the microscopist would take the sections on an ultramicrotome outside of the microscope and then bring them inside the microscope. With SBF SEM, you have the idea of just having the ultramicrotome inside the microscope and cutting out the middleman in that sense. So the way this technique works is you have your sample, you have an ultramicrotome net blade, and you have your scanning electron microscope. So scanning electron microscope, it scans across the sample, as opposed to a transmission electron microscope from the previous technique, which shines electrons through a sample. You have your sample, it is imaged, and then the sample is raised. The ultramicrotome comes and cuts away the top layer, and then the sample is imaged again, and you repeat that, and then hence you get your 3D volume of 2D images. But we've got a handy animation that's going to make it really easy, hopefully, to understand. It's been around conceptually since the 80s, but it only really became a big thing in the late noughties with Gatan's 3V machine, which is the machine we have here. So here's that animation. I love this animation. It makes it super easy to understand. So here you've got your sample, your resin. This gray area is the sample. Here's the SEM column, and here's where the images will appear. That's the knife. And very simply, image is taken of the flat block face, image is formed, beautiful stuff. The sample is raised, the knife comes across and cuts and repeat. The distance that the sample is raised between each cut is the slice thickness, and this is variable. So you might have a 50 nanometer slice in between each cut, or 100 nanometers or something like that. Importantly, the sample is destroyed in this process, whereas with the previous technique, if you wanted to, you could go back and image your slices if you cared to store them. With this technique, you cannot do that. They're just lost. They're destroyed in this process. This is our machine. This is the Quanta 250 FEG SEM, which is a relatively standard SEM. And when you buy a 3V from Gatan, what you actually buy is this door, this very posh door, which you fit to your microscope. Just slides in, fantastic. And here is where the sample is kept, and you can see here's the knife. A lot of shiny metal work inside. So this is just a view from inside the microscope. It's very cramped in there, but this gives a good idea of like what's happening. So panel B here, you can see this is like a line drawing of the red square. Panel C is the sample, and then you've got the knife holder here. Panel C is just showing the electron beam scanning across the sample during imaging. Then panel E, this is where the knife apparatus comes over the sample in order to cut it. And then panel F, the diamond knife, which is a very tiny part of the apparatus, cuts the sample and repeat. So yeah, we got our machine in 2010. It's had a lot of work since then. Runs take several days, and we've had a lot of runs in our time. So yeah, that microscope's been chugging along, done some great work. Back when we got it, there have been advancements in the technology since we got it, and there are more up-to-date versions that have basically they have technologies that allow them to get a better resolution and a better Z resolution, importantly, aka a thinner slice thickness, because a lot of the trouble with this technique is just trying to get the knife to cut properly. But that's something I have to worry about, not any users. So yeah, as a means of demonstrating the differences between this technique and improvements over SSTEM, this is from Wee in the Cadler lab. This is a embryonic tendon done both in SSTEM on the left and SPF SEM on the right. So you can see with the SSTEM sample, it's got the artifacts, etc. Whereas SPF SEM, you can see it's a lot smoother. The images are they're much more well aligned. The resolution is still really good by a lot of standards. Like you can see the collagen fibrils really easily, the crystal of the mitochondria, etc. The resolution isn't quite as high, but it's important to know that for a lot of applications, this resolution is more than enough, much more than enough in a lot of cases. So here's a data set that I took, which I really like, to demonstrate one of the strengths of this technique, which is the fact that it can image quite large volumes with relative ease. So this is from Eamon in Thornton lab. This is a cement gland of a tadpole. So we were able to image the entire cement gland, which is, like 250 microns cubed, 250 by 250 by 250 at quite high resolution. So these pixels are, I think, around 20 nanometers in size, which isn't as high as serial section transmission electron microscopy. But as I've shown with this image, it's more than enough to resolve membranes of the ER, lots of details, vesicles and so on, large vesicles. So some strengths and weaknesses of this technique. Strengths, it allows imaging of relatively large volumes quite quickly with a decent resolution. So typically that run is a bit bigger than we'd usually do. Often we do something like 50 by 50 by 50 microns at maybe 10 or 20 nanometers pixel size, and it might take as quick as 24 hours. It does have the poorest resolution of all the volume EM methods, but as I've said, it's still quite good. So yeah, this is a table that I've taken from a paper and modified. It just shows all the techniques that we'll be talking about today, but don't worry too much about absorbing all the detail. It's just good as a reference atlas for where we're going. So the next place we're going to go to is FIB-SEM. So FIB-SEM is a technique very similar to serial block-based scanning electron microscopy in a lot of ways. The main difference is that instead of using an ultramicrotome mounted in the chamber to cut the sample, you use a focused ion beam inside the chamber to mill away the sample. So this concept of using a focused ion beam in electron microscopy has been around for a while. In fact, it was first used to get around a certain problem. Often we might want to do electron microscopy on samples that are really hard, like maybe bone or dentin. These samples you can't cut conventionally on a transmission electron microscope on an ultramicrotome for a transmission electron microscope because they're too hard and they'll damage your very expensive and precious time in life. So what people used to do is, they still do it to this day, they use a focused ion beam to mill out parts of tissue, and then they remove them with a special tool, and they essentially have a special little slice of tissue that they've taken by milling with a focused ion beam. But this technique that we're talking about is the idea of in situ milling with imaging in an SEM. So what they have is, this is just a representation of what's in the chamber. So you've got this is the SEM column. Then to your left here, you've got the focused ion beam column, which has to come in at an angle. And essentially, you just mill away the sample in between rounds of imaging. I've got this little work diagram here that's going to hopefully help to make it clear. So first you start off, if you imagine when you put your sample into the microscope, you're going to want to like see what's there, plan your run, and like look at where you want to mill, etc. So the first thing you've got your flat block face. You put that in the chamber and then you image it with the E-beam, the electron beam, and map it out. Then you decide where you want to image, where you want to mill. Importantly, uniquely for this technique, the sample when it's being imaged is actually tilted because the beam mills in like a straight line. So in order to mill correctly, you have to tilt the sample. Some modern ones tilt the sample back for imaging, but it's not important to know. So then you mill away a big trench, and this reveals a nice flat block face similar to SPF SEM. And then you can image this block face with your SEM, mill, image, mill, image, mill, image, and what you're left with is a collection of 2D images representing the 3D volume. And then you need to align your slices. Software can help you with this. And uniquely for this technique, the software must correct for the tilt angle, obviously, because you're imaging at an angle, which can be quite unusual to get your head around when you see it, but it makes a lot of sense in the end. So this is a data set which I found recently. What I like about this data set is it shows how high the resolution is, and you can really clearly see the nuclear pores and loads of details about the cell. It's really pretty. A key advantage of this technique over the others we've spoken about is that you're able to, because focused ion beams aren't limited in the same way that ultramicrotomes are, you can mill away arbitrarily thin amounts of sample. So this means that rather than being stuck with 50 nanometer slices, your Z resolution is going to be the same as your X and Y resolution, which is really, really great and gets around the main limitation. One of the main problems with this technique is that it takes a long time, because unlike SPF SEM, where your knife comes across and does all the hard work for you, here you have to wait for the focused ion beam to mill things away, and it's quite slow. But yeah, so the resolution is higher than with SPF SEM. We're talking maybe 10 nanometers in X, Y, and Z, which isn't the best, but it's still relatively high. It's accurate, but it's slower than SPF SEM. And importantly, the more you image with this technique, if you imagine when you're milling away with your focused ion beam, you're creating a lot of contaminants in the chamber. So this means as the run progresses, it's prone to artifacts, which is a limiting factor. And the maximum volume you can do is around 20 by 20 by 20 microns, so significantly smaller than SPF SEM. Where where might you use this technique? So whereas with SBF-SEM, you might want to look at a whole cell or interactions between cells or a group of cells, or maybe the location and distribution of an organelle within the cell or something like that. With FibSem, you can't do things that big, but you might want to look at something that needs higher resolving power, such as the interaction between vesicles and the Golgi or something like that. That's going to be hard to resolve with the Z resolution of an SBF-SEM. So the next technique we're going to talk about is a broad family of techniques called array tomography. And array tomography is in use not just in electron microscopy, but in light microscopy as well. This technique essentially takes SSTEM is a good starting point to go about thinking about this. Whereas with normal SSTEM, someone has to manually collect every section, and then they take it into a TEM and have to image each section individually. The idea of array tomography is you do this in batch. So you have a machine that automatically collects the sections for you, and then you image all the sections simultaneously, and thus massively reduce the incidence of artifacts and increase the throughput. So we're going to take a look at AtomSem, which is the most popular method of array tomography in electron microscopy. So AtomSem started out life as Atom stands for Automatic Tape Collection Ultramicrotome, which should give you a good idea of what's happening. Essentially just a machine that you attach to an ultramicrotome that for both light and electron microscopy will collect the sections for you and store them safely. And then you can go about imaging these sections, in the case of electron microscopy, inside the chamber of a scanning electron microscope, and have software do the imaging for you, or a lot of it, and massively increase the throughput. So this is what one of the machines looks like. This is a special ultramicrotome. Anyone who's used an ultramicrotome will recognize this area as being very reminiscent of just a normal ultramicrotome. You've got your arm with the sample, the diamond knife here, and the boat. What's unusual is this huge big metal box that looks like it's got a reel-to-reel tape machine on it, and that's where the samples are collected and stored. Got a video here which I really like. It really demonstrates the technique well, and it gives you a good visual idea of what's happening. So yeah, I might interject at one point, but we'll have a go.
Excitement is growing around the AtomTome, or Automated Tape Collecting Ultramicrotome, an instrument that was originally developed by Harvard scientists to collect thousands of ultra-thin sections for imaging. Today, the AtomTome is being developed and commercialized by the RMC products team at Beckler Instruments. Now in the early adopter phase, RMC is working with highly innovative investigators who are interested in doing their research while helping the company refine the instrument, the associated protocols, and various potential correlative workflows. The AtomTome collects ultra-thin sections immediately after they leave the diamond knife, depositing them on a spool of Kapton tape. To give you an idea of the delicate handling required in the process, these sections...
Just interject here, if anyone's not used an ultramicrotome before, this is what you'll be looking at when you've got through that, the binocular microscope. So here you've got your sample being held on the end of the microtome arm. This thin line here is the valuable diamond knife. And these little gray sheets are slices around maybe 50 to 100 nanometers thick around that area. And that's what you'll actually be having to preserve an image. And you can imagine why this is so difficult to do manually, and why this AtomSem helps so much.
Are only 30 to 50 nanometers in thickness, about 1/1000th the thickness of a human hair. After the sections are collected, researchers then cut the long rolls of tape into shorter lengths and mount them on silicon wafers or glass slides. These populated substrates are then ready for staining, immuno or fluorescent labeling, or coating. Resulting images can then be used in array tomography to reconstruct 3D structures, or the images can be reviewed to identify and localize regions of interest. Most importantly, the sections can be retained for years for further imaging. The AtomTome's unique ability to collect hundreds to thousands.
There we go. I love that video. Fantastic accent. You've got the classic reference to the thickness of a human hair, which is obligatory in all the EM videos. So your workflow for this would be something like you have your staining and your embedding in resin, and then you have your automatic tape collection. So that requires some skill to set up the ultramicrotome. But once you've got it running, you can collect a lot of samples, a lot of slices of your sample. Then what you want to do is cut up your tape and put it on something that can fit inside an SEM chamber. So here you've got lots of ribbons of tape, each with sections on a silicon wafer that can be imaged inside the SEM. You load the wafer into the SEM, and then you take your map image and figure out where you want to image, like, oh, I'm interested in this structure that appears here. And then the software will help you out and map that to all the other slices. And then you press go, leave it, and in a while you'll have your images. And depending on how big the volume is, you might have to do many different wafers. So this is a data set, a recent data set that I found. What I like about this, as well as showing you that the resolution is quite high, you can see the double membrane really clearly of the nucleus. You can also see, if anyone's ever imaged with a slightly old diamond knife, they'll recognize these little knife marks here, which shows that this technique is prone to the same errors. And here's what they've done with it. So this, I love that this shows the volume in 3D. So there you go, that's the data set. Yeah. And what they've gone on to do is, create a fantastic model of the mitochondria, ER, and so on. And this is the type of thing that you might want to do with all your volume EM data sets, depending on what question you're interested in. They actually used their own custom machine learning algorithms to model this, because modeling can take quite a lot of time. So yeah, it's very impressive. Big advantage of array tomography and AtomSem is allows you to image huge areas, because when you've got your samples in the SEM, you can tell the computer to take a series of images per slice rather than just one. So you end up being able to possibly image, like, we're talking millimeters. That'll take you a long time, but it's possible. The sample, you get to image the slices later on, as the American person told us. You get to store them safely. The resolution in XY is comparable to FibSem, which is very good. And the Z resolution is analogous to the other slicing-based techniques such as SBF, SEM, and SSTEM. And it requires expertise. And also, ultimately, at the end of the day, you're still limited by the fact that you only have one beam in your electron microscope. So it takes time. Unless you have £5 million to spare and you want to get yourself a brand spanking new Zeiss Multi-Sem 505. So this is a relatively new technology. It came out in the last couple of years, I think. Still not available to the general public, I believe, but the crux of it is that you have an SEM that has many beams that image in parallel, which to me just seems fantastically futuristic. I'm not quite sure how it works. I would love to know. And each of these beams is detected independently with its own detector path, they call it. Don't quite understand it, but it shouldn't be hard to see that this could massively increase your throughput to the order of, you would end up creating apparently several petabytes of data a day. That's a lot of hard drives that you'd have to carry in and out of work. So yeah, if anyone wants to lend me 5 million, I'd be very grateful. So that's AtomSem. The next techniques we're going to talk about are slightly different. They're going to be tomography techniques. So tomographic techniques are different to the techniques we've spoken about before. I would classify the techniques we've spoken about so far as being volume EM techniques. And what I mean by that is they're all based in some form or another of taking images of physical slices through the volume. So your data set of 2D images is actual images that were taken by the microscope. Whereas with tomography, there is no sectioning or cutting. What you have is a 3D EM data set that is generated mathematically or computed, and it's computed from a set of 2G projections through a 3D object, which sounds very abstract, but it's actually not too difficult to understand. You don't have to understand the maths behind it. I don't, but it's easy to think of how it would work. So the mathematical concept of tomography has been around for about a century, and it was first used to map our solar system, I believe. But since then, it has become very important, not just in EM, but in other fields as well. One of the great things about tomographs is I know many of you will have featured yourselves in your very own tomographs, and that's because tomography is the technique used in X-ray CT scans. So X-ray CT stands for X-ray computed tomography. And it's a great way to understand the base of the technique. So what you have is, in this case, X-rays, but the comparison to electrons is easy. You have your X-ray source here, and then it's passing through this grumpy bald man's head onto an image receiver, a camera most likely. And if you look at the other side, you can see what you can see. You have these X-ray type images, and these are basically projections through this person's head at different angles. Obviously, it doesn't quite occur like that in an electron microscope, because I can't imagine how expensive it would be to build a microscope that can rotate in 3D around the sample. So what they do instead is they tilt the sample inside the microscope. So you have your electron gun. This is a transmission electron microscope. So the electrons are being shone through the sample and the image is collected at the bottom. The sample is rotated, is tilted, and then here you've got your image collection at the bottom. And what I like about this technique is you don't have to understand the maths to kind of be able to just understand that you should be able to get 3D information from it. It just seems to make sense. And here's a more diagrammatic representation. You've got your electron beam here on the left going through the sample, which is being tilted. On the right, you can see these projection images, I believe, are taken into Fourier space, combined and taken out again, and some maths happens. But all you need to know is at the end of it, you get your 3D data set, just like the data sets we've been talking about, obviously a lot higher resolution. I'll show you that it's higher resolution. This has been done in biological transmission electron microscopy for a long time. This is a paper from the 60s, I think, where they used a very rudimentary version of this technique to model the bacteriophage tail. And they've made the model out of plastic, which is something you don't see these days, but it's still very cool. So one of here's a data set, a recent data set. I want you to look at, firstly, you can make out the Golgi really clearly over here, which is really cool. And it can be hard to, because it's so high resolution and the magnification is so high, it can be hard to even place yourself if you're used to looking at images of cells. But what we've got here is the mitochondria in purple and the ER in blue. So they were able to create really high resolution maps of this. One thing to think about with this technique as well, that's obvious from this volume, is that you can only image one slice at a time. And so you're limited to the thickness of your slice because the electrons have to be able to pass through them, as with SSTEM. So the depth of your volume is severely limited to something like, 100 to 200 nanometers, which isn't a lot of space. You're also limited by your field of view in XY as well. So this technique is not great for imaging huge things, but for imaging tiny things, you can't get better. So yeah, this is the highest X, Y, and Z resolution 3D EM technique. Whilst your Z depth is very low, because you're reconstructing this with tomography, your Z resolution is no different to your X and Y resolution. But as I've said, it's only useful for very small, shallow volumes. You also have a unique artifact with this technique called the missing wedge artifact. And this is essentially derived from the fact that, unlike in an X-ray CT machine where you can scan your scanners around the person 360 degrees, you can't do that in an electron microscope for physical reasons. So you can only tilt your sample to maybe 80% of the way you'd need to on a good day. And that results in some missing information. So this diagram is just representation of if you put Albert Einstein in your electron microscope and did the tomograph of him, how much information would you miss if you were to have this missing wedge artifact? And you can see that further to the right, you've got this kind of unique visual appearance that is quite recognizable, and the information gets of poorer and poorer quality. This also shows that 5 degree and 2 degree increment, that's the increment between images on the tilt. And you can imagine that the bigger your increment, the less complete your data is. So if you've got a smaller increment, that increases your data quality. But there are various questions that come into that, like the more images you take, the longer the time is, and the higher your dose is, and your electron dose can damage your sample. That takes us on to the final technique we're going to speak about, which is cryo-electron tomography. So this technique is a tomographic technique, so it's entirely analogous in that sense. But it has a unique feature that's very different to any of the techniques we've spoken about so far, and that's the fact that it uses cryo preparation techniques. So essentially, instead of using samples that are stained with heavy metal and embedded in plastic, this technique uses samples that have been frozen. And there are two ways you can go about this. You can freeze normal tissue. You can freeze small amounts of tissue and then high pressure freeze them, and then section them on cryo ultramicrotomes, which are special microtomes built for the sectioning. And then you can image those sections, or you can image vitreous ice. You can essentially get concentrations of the thing you're interested in, whether that be a fraction of a cell or a organelle or a virus, for example, and then you freeze this in vitreous ice using plunge freezing, which we're going to cover in a second, and image the film of water with your sample dispersed throughout it. And this technique allows for a futuristic, very cool thing called subtomogram averaging, which we're going to talk about in a minute. So this is your workflow for the vitrification method. So this is for when you have, say, a suspension of viruses in your water or something like that. So what you do is you get your standard EM grid, you put your a small amount of your sample on it, and what you'll have is a drop of water. You can't image a drop of water, that's much too thick. What you want is a very thin film of water, and the way you achieve that is by using blotting paper. So you blot away the excess water, and what you'll be left with is an EM grid where the holes have, like you can see here, a very thin layer of very thin layer of vitreous ice. And to freeze it, you plunge it into liquid ethane. It's really important that you plunge it into something incredibly cold, like liquid ethane, because if you freeze it normally, if you were to decide that you couldn't be bothered to use your VitroBot today and you're going to put them in the freezer, what you'll get normal ice forming, and normal ice has a crystal structure which is very detrimental to imaging. So you need to freeze your samples so quickly that the ice doesn't have a chance to form a crystal structure. And then you image the sample in the holes using the techniques that we've talked about using tomography. So here's a data set I like. This is obviously really hard to make out where you are because the magnification is so high. But what we're actually looking at is the crystal of a mitochondria. So they image some mitochondria using a vitrification method. And what they've been able to do is get this here. This is a section of crystal with the ATP synthases. They're the yellow molecules. So when I mentioned the technique of subtomogram averaging. So what you can do is, if you have something like this where you have a repeating kind of motif or complex that appears many times in your tomograms, you can computationally average them out and to create high resolution models. So here they were able to create a 14.8 angstrom model of ATP synthase. So anyone who's done scanning, who's done single particle, will be scoffing at such a ridiculously low resolution. But I really like that it shows that on the one hand, with the 3D electron microscopy, you have techniques that don't do great resolution, but can do large volumes. And then on the other hand, you're getting seriously close to single particle. And if anyone hasn't seen, I really recommend Rich's talks on Rich and Mike's talks on single particle, because they're very informative and show you what that kind of maximum resolution that people are getting these days with proteins, which is seriously cool stuff. So yeah, that's cryotomography and tomography. Ultimately, the highest resolution in X, Y, and Z, but you're limited to the size of volumes you can do. Cryo-electron tomography has its own problems because in conventional electron microscopy, the heavy metal stain negates a lot of the problems you might get with charging, whereas carbon is very prone to being absolutely ravaged by high energy electron beams, and as a result, you're limited to small doses. The signal to noise can be very poor. But ultimately, it allows you to render really small stuff and can be super good. So for the last, how long have we got? Maybe five minutes. I've just got a few kind of twists and spins on the techniques we spoke about today, because like single particle 3D electron microscopy is a field that's always advancing, and people are always thinking of novel applications and modifications to the techniques. And it'll be really interesting to think about where we're at in 10 years with some of the stuff that's happening. But first, this technique is called 3D emish, and it's something you use in SBF SEM. So this technique uses DNA probe hybridization to specifically target sections of DNA with gold. And gold, if anyone doesn't know, gold provides contrast in an electron microscope, so it's useful for targeted studies. So what they've done, they've identified the regions of DNA that they're interested in, stain them with gold, and what they want to see is during mitosis, what formation do these chromatin structures take? And this is something they've not been able to work out in other ways. So what they've done essentially is stained these specific chromatin structures, and then when they've gone and sectioned with an SBF SEM, they've been able to get this really nice data set. Whoops, a little spoiler. They've been able to get this really nice data set these chromatin structures, which is really cool and something I didn't think, an application I didn't really think of until researching this. So now we'll move on to a couple of FibSem techniques. So if anyone remembers, one of the main limitations I said with FibSem is that you're really limited to the depth of sections that you can image to 20 microns. And, you know, sometimes you're going to want to image a lot more than that. You don't want to be limited by such a small, relatively section size. So what this technique is, it kind of combines the concepts of FibSem and also just classic serial sectioning. So you have, it's called hot knife sectioning, and it involves a knife that's been heated, not to a huge heat, something like 40 degrees, I think, and then it's vibrating ultrasonically. And this allows you to cut much thicker sections than you would conventionally be able to cut, and sections as thick as 20 microns. And what you do once you have your 20 micron serial sections, you image each of them in a FibSem in series, and then you can combine this data to create really nice big volumes at FibSem resolution. The group that have done this have a lot of FibSem. So what they did was they put each of the sections in a FibSem and imaged them together. Obviously, if you've not got a lot of FibSems, then it's going to take you a long time, but it's still a super cool technique. Another one, it's a FibSem. So this is kind of a combination of FibSem technology with array tomography. So if you remember, in array tomography, what you have as part of your process is inside your SEM chamber, you have a wafer, and that wafer has lots of slices on. So with gas cluster ion beam technology, instead of imaging thin slices once, what you do is you put your thick slices that may have been collected on a hot knife sectioning machine, for example. These 20 micron thick slices, you've got an array of a few dozen of them on your wafer. And you use like a FibSem technique, but instead of a focused ion beam, you use a cloud of gas ions to mill away all of the sections simultaneously. And I don't know if you remember, one of the main limitations with focused ion beam scanning electron microscopy is the time it takes as well. So you massively reduce the time that it takes, and allows you to essentially image an array and also mill as an array, which massively increases your throughput. And this is compatible with multi-beam SEM technology. So when we get that £5 million, we'll be rocketing through. My last technique, I really like this technique because it's very much, it's not so much technologically driven, it's just hard graft. So this is the concept of tomography stacking. So if anyone remembers, problems with tomography are that it is super thin. It's very thin sections, 100 nanometers, 200 nanometers, something like that. But it has really high resolution. Often you need to image, you need to make models of things that require high resolution, but you don't want to be limited by the thinness of tomographs. So what they've done here, and this technique has been around for a while, I think, they combine the ideas of serial section transmission electron microscopy and tomography, and they have essentially stitched together serial tomograms. So they collect their slices, image them in series, but image them as tomograms, and then they recombine them into one. So what they've been able to do here is create structures of the mitotic spindle in different stages of cell division. They've recreated the spindle in different organisms, and the spindle, you need high resolution. This technique is, I believe, quite a pain because you have the same, some of the same problems that you have with serial section transmission electron microscopy, in that you have to kind of manually align things. And I know in this paper they had to do a lot of making sure each little bit of the spindle was attached to the right place. But the end result is super impressive. And I really love these figures that just show you, like, really clearly these are tomograms stacked on top of each other. And yeah, that's a whistle-stop tour of 3D EM. Yeah, the main techniques we do here, we do a lot of 3D and we do tomography as well. But if anyone has any questions about any of these techniques, or any inquiries about 3D modeling, or just wants to chat about anything, please feel free to get in touch. We can have a chat. Yeah, I hope that's been informative for everyone. If anyone has any questions, please, yeah, leave them in the chat.
Thank you, David. It was a brilliant presentation. It was really interesting. I like the last part because actually expanded a bit of my knowledge as well. We have not a lot of questions, but one is quite interesting question from Priya, if I'm correctly saying this name. Sorry for naive question. How exactly we differentiate between different cellular components from tomography results, as they are all negatively stained? Actually, they're not negatively stained. And for tomography, we're using standard routine TM preparation, which involves positive staining. So actually, most of the structures, they looks like dark material on a transparent or white background. So there is no problems in differentiating. If you know the shapes, of course, and recognize patterns, there is no problem. There are no problems in recognizing cellular components. And for particles, I think tomography are not used for negative stain particles in most of cases. They're used for cryo preparations sometimes, especially when particles have preferential orientation or they are unique particles. Then you can't have a lot of projections of different orientations, and then tomography will help a lot, really. Next question from same person is, any labeling coloring technique to label parts of specific RNA you are aware of? Actually, I'm planning to check if my RNA of interest become circular after stress, like shown in this paper. Labeling for EM is usually done with, as David said, with electron dense markers like gold particles, colloidal gold, some other variations, but then kind of minor. And of course, it depends very much. If you're using fish technique, not fish, like EM-ish technique, it means that everything is dependent on how good your probe against a certain place of RNA is. And there's a question, can you recognize RNA itself? And I think here we can kind of hit the wall of resolution for routine TM preparations, because the preparation and contrasting techniques, they are limiting AM resolution to about 1-2 nanometers, depending on how good the things are. And of course, you will not see the lines clearly of RNA. You will maybe be able to label where this specific sequences of RNA are located, but you will not see RNA itself with a routine TM preparation. I think there is a question for David about this SBFM. Serial block phase SEM from Michael Murray. Great. No, you can read it probably, David. It's great talk, David, about a serial block phase SEM. Is there a way of correcting discrete misalignment along the Z-axis in the stack? That's for you, David, I think.
Yeah, so as in like errors between like the ways the slices are registered. So when you're traveling down your volume in the video, you see like maybe jitter or that things don't quite look like they're in the right position. There are ways of doing that. We use software, so you can either do it manually, like yourself deciding that I want to move it somewhere different. Obviously that introduces could be a source of bias. There are also automatic software options of doing that, and a lot of the time when I'm creating SBF SEM datasets, I do apply some kind of protocol to just correct slight misalignments, because it's a physical technique with a lot of moving parts, and so there are inevitably some errors. But if you have any data that you are looking at and you think, oh, I could need to something's misaligned, just we can chat about that, because we have many ways of fixing that.
There's an interesting question from Caroline. Is there any way of using archival EM specimens? This would open up many opportunities. I think the whole field of these techniques, they're moving towards that because you have better and better signal to noise ratio, especially with the recent invention of focal, I think, charge removal.
Yes.
The main problem with the samples in most of these techniques, especially when you use resin-embedded samples, that in SEM you need conductive sample, and resin is not conductive. Means you accumulate charge, and to remove the charge, you need to introduce some water vapor, and then it becomes kind of low pressure, low vacuum SEM, which actually decreases the resolution. And for this new suggestions, new methodologies like focal removal of the charging, you do not need any water vapors there. So you can create much better vacuum, meaning much better resolution. And it means as well that you don't need very heavily stained samples like for standard serial block phase imaging, meaning again, we can probably at some point do this archival EM samples, but not in our arrangement, because we don't have this focal charge compensation. But I think if you will get new machine, yeah, at some point, this will improve a lot of imaging. I think Alan asked about rotary shadowing for visualization of DNA and RNA. Yeah, of course, it's possible, but then it will be just flat, and there are a lot of different things, like different peculiarities and artifacts for this rotary shadowing, and it could be just done on a very flat surface, and of course, shadowing itself decrease the resolution. It can show shape, of course, but then you need to have very clean sample, because anything you put in shadow, it will be shadowed. So any garbage around, you will see it, and it can obscure the real sample. But if you want to discuss all the options which we can think of, then please come to us. Of course, in COVID times, you maybe not be able to come personally, but email, telephone, our Teams, Microsoft Teams is a great place to discuss things, because then we can share all our knowledge between all our members. So don't have probably any more. Then I again thank everybody. Here's a new message. It's again thanks. Thank you for thanking. Thank everybody for attending our series of lectures, and I really enjoy to be a host, and to give an opportunity for David to deliver such a brilliant presentation.